Hi everyone. It's so great to talk to you via Zoom. This is amazing. So right now I am in Lyon, which is a small city at the base of the French Alps. So it's a beautiful city. We're very well known for our Festival of Lights every year. But we're also known for the Centre Lyon Brad, which is a cancer research center that's based here in Lyon. So here we research everything to do with cancer, and our team specifically is interested in understanding the epigenetic drivers of hepatocellular carcinoma. So when I talk about DNA methylation, we're talking about it in a CpG context. So overall, our genome is highly methylated, with over 90% of CpG dinucleotides being methylated, with the exception, of course, being in CpG islands. And so usually these occur in promoters and enhancers of genes. And when we do see methylation in CpG islands, it's highly correlated with gene silencing. But as you all probably know, the picture is more complex than just methylated cytosines. We also have hydroxymethylation, 5-formylcytosine. We have other modified bases that can occur in different regions in the genome. And so when we look at, for example, hydroxymethylation, so this is probably the second most well-studied modified base. And usually we see it concentrated in the enhancers and the bodies of actively transcribed genes. So a very different profile. And then when we look at liver disease, so usually we're talking about hepatitis, which then often moves on to cirrhosis and usually to the hepatocellular carcinoma. We usually see global hypomethylation in this biological context. And so studying DNA methylation is really important to try and understand the mechanisms of disease progression. And also it's useful for developing novel biomarkers. So if we look at, for example, occult HPV infection, we don't really have any good biomarkers for this disease. And so using DNA methylation, it offers a potential stable biomarker for measuring occult infection. But when we take a closer look at our liver cells, we have nuclear DNA, of course, which has been very well studied, where we know methylation occurs. But we also have other cellular subunits where we can also have DNA and also have methylation. And so when I'm talking about this, I'm talking about mitochondria, where we have DNA in this circular structure, and also viral particles, we can have DNA. And so studying these in the context of liver disease is very important, and it also can be very challenging with traditional sequencing techniques. So I'm just going to move my window over here. So as I introduced before, we know that DNA methylation in the nuclear DNA is highly correlated with transcriptional silencing. But when we look at specific genes, so for example, we published a paper in 2017 in Stem Cell Reports, where we showed very nicely that methylation at a specific gene, for example, H&F4A, changes here can be a driver of differentiation of liver cells. And even more recently, we published a preprint this year where we showed that hydroxymethylation can be a marker of actively transcribed actively transcribed genes in the liver during differentiation. But there are some problems with sequencing DNA using traditional sequencing methods, and the biggest one for us is that repetitive elements, or these junk DNA, they're not properly annotated with short reads. And usually we can see a lot of methylation changes during differentiation and in disease in these regions, but it's difficult to detect. Also, detecting DNA modifications at all in the nucleus is quite difficult because we rely heavily on things like sodium bisulfite conversion to look at methylation. And even we have to modify our DNA even more to look at other marks like hydroxy by doing oxidative bisulfite conversion, for example. And so for mitochondria, the liver is a highly metabolic organ, so it's not surprising that mitochondria are important in liver disease. In fact, there was a nice paper out of Italy in 2017 where they showed that when we have mitochondrial dysfunction, it's important for maintaining a viral infection. And when we don't have this dysfunction, the virus is more likely to be eliminated. And even in our own hands, these pictures at the top here, I don't know if you can see my mouse, but when we look at the liver cells, liver progenitors, and we compare them to differentiated liver cells, we know that the number and activity of mitochondria increases a lot in differentiation of these liver cells. But there is a lot of controversy surrounding whether or not mitochondrial DNA is even methylated, and so this arises from some studies where they had difficulty in doing a complete bisulfite conversion and improper linearization prior to sequencing, and so this led to some erroneous conclusions. And also mitochondria, it can be quite difficult to sequence them with traditional techniques because the sequence is highly variable. So in the same cell, you can have different mitochondrial sequences, and this can be very difficult for something like developing primers. And then lastly, we have viral DNA. So HBV is a virus that infects liver cells, and even though we do have a we do have a vaccine for this disease, there are still 257 million chronic carriers of HBV, and so they can still transmit the virus by vertical transmission. So it's still a big problem. And there was a nice paper in 2009 where they showed that with HBV within the virus, that methylation of the virus can be involved in silencing the viral transcripts, so HBS, for example. But because of its unique structure, so it's a 3.2 kilobase relaxed circular DNA structure, so it has this strange gap, it can be quite difficult to sequence with these traditional techniques. And in addition to that, it's a highly mutagenic virus. So there's lots of different genotypes and it has highly variable sequences. So once again, difficult to design primers. And as well as that, viral combinations can be very difficult to detect with short reads. So why nanopore sequencing? Like, I'm sure you're all converts, but for us, nanopore is really, For us, nanopore is really important because we don't need to do PCR and we don't rely on bisulfite conversion. And so this is revolutionary for people working on modified bases. The protocols are pretty convenient, some more than others. We get these super long reads. We can look at allele specificity. I'm sure you guys all know this after your very long week of workshops. But for us, we can detect these DNA modifications. So Jared Simpson and Winston Timp in those two teams, they developed nanopolish, which is what we've been using. But you've got the new Guppy and even more modern techniques to be able to sequence modified bases. So the first thing we really wanted to do was we wanted to look and see if nanopore was telling us that a cytosine was methylated or hydroxymethylated. So we know that by using sodium bisulfite, sodium bisulfite conversion, we can't tell the difference between methylated cytosines and hydroxymethylated. And so we wanted to be sure that we were seeing this distinction with nanopore. And so what we have is these synthetic standard DNA, so where every cytosine is either methylated, unmethylated, or hydroxymethylated. And so we just did the rapid barcode sequencing protocol. We sequenced for 48 hours on the mini-ion flow cells with the chemistry 9.4.1. We use Guppy for base calling, nanopolish for indexing. We align with Minimap2. We do nanopolish for methylation calling, and we do allow our statistics and our graph visualizations in R. And so when we plot the methylation frequency and compare the unmethylated to the methylated, we see this huge difference. And we were very happy with this, that it was beautifully showing us that these cytosines were methylated or not. And with hydroxy, we saw something different again, which gave us a lot of confidence that we were detecting methylated cytosines in the absence of hydroxy. So then we wanted to look at nuclear DNA methylation. So whole genome methylation sequencing was what we looked at next. And so I'll show you some data on our cirrhosis patient cohort. So we have, it ended up being about 300 patient liver samples where we had cirrhosis patients from various etiologies, and we also had normal and liver tissue matched pairs. And so when we were setting up this experiment, it was going to take me about 4 months to sequence all these samples with a mini-ion just to do whole genome bisulfite, whole genome methylation sequencing. 4 months it was going to take me, doing library preps 3 times a week. It was going to be crazy. So we set up, I was very lucky to meet Kevin Lebrigand, who is working at the University of Côte d'Azur genomics platform in Nice, and they have a Promethean. So I set up a little in parallel experiment where we were able to test the efficacy of whether or not it would be useful for us to use this better, bigger equipment. And so when we compare the library preps, they use the same kit, The only real difference for us was the number of barcodes we used. So with the Promethean, obviously, it has many more flow cells, many more pores per flow cell. I think it's like 7,000 compared to the mini ion, which is about 1,000, 1,500. And so we use 24 barcodes versus the 8. And the runtime is slightly longer with Promethean and the volumes are slightly different. But other than that, the library preps are pretty much the same. And when we look at the data output, so this is data from your everyday user. We're not working in the Oxford Nanopore technology labs. So this is like pretty general what you would get. So for a mini ion flow cell, we got about 13 gigabases of data. And it would have taken us, like I said, 4 months to sequence everything. And so with the Promethean, we got 50 gigabases of data per flow cell, which was great. And we were able to sequence all samples in just 2 weeks. So this was incredible for us to get this crazy amount of data in such a short amount of time. But it's important to note that we did have a much lower N50. So with our Promethean runs, we had an N50 of about 6, but with the mini ion, it was about 16. And so we think this is probably because we had to send the prepared libraries, and they weren't loaded immediately, but we're really not sure. But this is pretty normal from what we've seen. And so then when we look at these graphs here, once again, don't know if you can see my mouse, this is showing us the average, the absence of methylation in CPG islands. So, whoops. So here we have a CPG island and then we have the shores either side. And so we expect, this is a nice quality control where we expect to see this absence in the CPG islands and not in the shores. And so this was showing us really nicely that regardless of the device, as we expected, we'll still be able to detect methylation in these liver patient samples really well. But we want to look closer. And so whole genome sequencing is very useful for lots of things, but we're really interested in looking at specific regions. And so I'm going to switch to a different model here where we differentiate liver cells in vitro. So this is, we seed progenitor cells and then they differentiate into mature hepatocytes in vitro. And so despite getting a good coverage of each chromosome, so this is showing the chromosomes here, we have 15x in some CPG sites and we have this bimodal representation of methylation as you expect. When we look at a specific region, for example, HNF4A or hepatocyte nuclear factor 4 alpha, so this is a transcription factor that's very important for the differentiation of liver cells. And so we've studied this region very closely and we know that we expect to see specific changes in methylation along this gene during the differentiation of these cells. And when we did our whole genome sequencing with Nanopore, we saw differences, but they weren't quite right. And so this is showing you the pattern in the proliferative cells and the pattern in the differentiated cells. And so I guess this is a really good example of when you need more depth to make conclusions about methylation on a specific region. So we had maybe 5x, maybe less of this region. And even though we did see differences, you really can't conclude anything from such a low amount of coverage. You really need much more. So we were very pleased to get our hands on the targeted sequencing protocol where we use Cas9. So for those of you who haven't used this protocol, we use Cas9 guided RMPs to specifically cut out your region of interest after blocking all the ends. So you only ligate adapters to your region of interest and you just sequence that region. And so Gabriele et al., they published a paper in 2018 where they use this technique to sequence the BRCA1/2 gene in breast cancer very nicely. And so the library prep is much more labor intensive. For us, we did single cutting. We had some problems with the double cutting and the tiling approach, but that's a different story, I guess. You ultimately use a ligation sequencing kit and you load 5 micrograms of DNA onto your flow cell. So another important consideration, because we're working with tissues usually, is it's difficult to get 5 micrograms of DNA. But if you can, this technique was lovely. And so we use all the same bioinformatics. We use Guppy for base calling, nanopolish for indexing and methylation calling, and we align with Minimap too. And we got a very nice enrichment. We got about 1% of all of our reads falling within HNF4A. And so this was very nice. We did get a lot of off targets, but mostly just from unspecific chopping. But when we look at the specific coverage of HNF rate was very nice. We had at least 100x along all regions, which was beautiful for this 40 kb region. So when you look at the methylation profile of this gene, so this is the HNF rate coverage, and then this is the methylation. So each dot is a CpG site, and along here we have our beta values. So between 1 is fully methylated and 0 is unmethylated. And so we have a lot more information to conclude about what's going on in this gene in terms of methylation in, I think this is in primary human hepatocytes. So we were very successful with the Cas9 technique. So we were excited to use it for everything. So we go back to the hepatitis B virus example, where this is a circular DNA structure. And so when we do whole genome sequencing of HBV infected samples, we're unable to usually sequence the virus without doing some sort of enzyme digestion because it's circular. And so we used the Cas9 guided RNPs. We used 2 to cut and linearize the virus prior to sequencing. And so in a model like this was in HBV infected primary human hepatocytes, we get a couple of 100 copies of the virus per cell. And so, yeah, that's an important note. So when we look at the coverage of HBV, it was crazy. We got around 15,000x for some regions, and which we were so happy with. And so when we look at the strand specificity, though, we do have a big difference because we have lots of replication intermediates. So the positive strand, we have this gap, and on the negative strand, we have these full transcripts. And so this was very nice. We were able to get a lot of data on HBV and a lot of coverage to be able to sequence this virus. So then when we look at methylation, there's only been a couple of papers that have been published that have looked at the methylation of the hepatitis B virus. And so for us and this particular genotype, we expect to see methylation around CpG island 1. And so that is here, this first green box here is the first CpG island. And so this was very promising when we saw this peak in methylation around this region and nowhere else. So we could clearly see that we're able to detect methylation in a virus using this technique, which was very nice. And we're also, interestingly, able to detect the absence of methylation around the other regions because we had such an excellent amount of depth. So when we look at mitochondria, we did, even though we had all this success with the Cas9 approach, we wanted to try and get a bit more bang for our buck with our flow cells. So we used a different technique. And so for this one, we enriched in mitochondria prior to sequencing. And so we used just the average lab-based equipment, like a Dunce homogenizer and the centrifuge, where we centrifuge the different gradients, and we linearize the mitochondria with an enzyme called BAM1 prior to sequencing. And so when we do it this way, we can multiplex a lot more samples on one flow cell. So with the Cas9 technique, we can do maybe 4 or 5 samples with multiple target regions. But with the rapid barcode sequencing protocol, obviously you all know we can multiplex much more and ultimately load less on our flow cells and get more use out of one flow cell by sequencing more regions. So we load 400 nanograms finally. And when we did it this way, we got a huge enrichment. It was crazy. If you look at the coverage plot here, just obviously in IGV, we got so much mitochondrial DNA that we were able to sequence from, this is just one sample. And when we look at the enrichment per chromosome, we had a huge enrichment in the sequences that were aligned to the mitochondrial chromosome. And when we look at methylation of mitochondria, we saw methylation, which was very promising, but we saw it specifically in certain areas. And so we were very confident with our positive and negative controls that we also use for this type of sequencing, that what we're seeing for mitochondria is real. And so while we didn't see any huge differences between the progenitor cells and the differentiated hepatocytes, which is what we're looking at here, we were still very confident that what we're detecting is really methylation. But it's very important. There's been a couple of papers published recently that have identified non-CpG methylation in mitochondrial DNA. And so this is what we want to move on to next. And I think that that's where we're really going to see when we develop the right samples to do the right, to be able to detect non-CpG methylation, it's going to be very important. So that's it for me. It brings me to my conclusion. So we are able to reproduce global patterns of DNA methylation in human liver cells, human tissues, human viruses. The protocols are simple. Some are more convenient than others. You're able to detect low but consistent DNA methylation in mitochondria. But we really need to do more to detect the other modified bases like hydroxy and non-CpG methylation. But to do that, we need a lot of bioinformatic work, and that is being done. We've got collaborators all over the world, and if anybody here is interested in working on hydroxymethylation, or if you have already been able to detect it, please let me know. We are always eager to collaborate. And yeah, and then I just need to say my thank yous. I'd like to thank you, my boss, Hector Hernandez, and mentor, and Isabel. My other collaborators in our lab here, Jeanette, Damien, Anel, Ariane, who is our bioinformatician, our other lab team, Julie Marie, and all of the people working with us, the University of Costa Azul Genomics Platform, Rainier Waldman, and Kevin Lebregon and Pascal Barbie, and all of our funders for the ANRS and the La Ligue Institute. And thank you all for your attention.
Thank you. Questions?
Very, very biology based. I'm sure you're all very tired, and it's been a big week by the sounds of it.
I had a question. So, because you did all your methylation calling with nanopolish, have you compared this to any other methylation caller? Do you know what?
Yes, we are in the process of comparing it to the new Guppy. So that's what we're doing right now. Yeah, because we think it'll be better with the new Guppy. Basically, there's some limitations with nanopolish, especially because we're interested in looking at strand-specific methylation. And so it's going to be easier to do that with the new Guppy than writing our own script and trying to do that in our own hands. Yeah.
And the new Guppy is like it's direct methylation calling. That's what you're referring to.
Sorry.
Are you referring to the direct methylation calling in Guppy, or?
So we're going to try that, as well as I'm not the bioinformatician. I wish I could answer it with more confidence. But as far as I understand, we're going to be using the direct methylation calling in Guppy. We're going to be using the same Guppy package, but we're going to be rerunning all the data that we've already called with nanopolish on the new Guppy.
All right, cool. Thank you. Any final questions here? So another question then. So for your Cas9, so you get like 1% of the reads are on target, and so the rest is mostly like off-target cutting sites of the Cas9, or? So
we haven't done much. So a lot of it is just fragmentation. So when you block all your ends, you obviously just lose some of those blocks just by handling your DNA, and so some of that DNA gets ligated and you sequence it. But in terms of actual specific regions where we get more than 100x, it's much lower than what that 1% on target value would let you believe. But yeah, I don't have exact figures for you at this time, but it's definitely something we're working on. There's the new, Oxford Nanopore have provided the ONT tutorial for Cas9 targeted sequencing, and so they give you beautiful plots where they show you all the off targets and the genes with reads of more than a certain percentage, but we haven't actually done those calculations. We only looked at our target gene for H and FRA, but it's something that we have to do for publication, so we're working on all of that at the moment. But yeah, that's a good question.
Do I have another question? Can you barcode the Cas9? So can you have multiple Cas9 cuts on the same flow cell?
Yes. So you can either look it up to, I think they recommend the maximum is 400 target regions of interest. So that's with one sample, you can look at 400 regions of interest. So 400 cut sites, 200 different genes. But you can only multiplex a maximum of 5 samples using the Cas9. So 5 samples with 200 targets, for example, is what they recommend with the nanopore guidelines. And so the most we've done is 4, because otherwise you're just loading way too much DNA. If you think about 5 micrograms for each sample, that's 5 to 15, 20, 25 micrograms of DNA, and it's just like trying to swim through soup to get to the pores to be able to sequence. So you decrease your throughput a little bit when you do it that way. But the flongles are really useful too. We just got flongles yesterday, so we're so excited to test the targeted sequencing protocol and those samples with flongles to see if it's useful, because that'll save us a lot of money than having to do 1 flow cell every time for just 4 samples.
Cool, thank you. Good luck with this. There was another question in the back. Yeah, so I'm gonna repeat this. So you mentioned something about hydroxycysteines, and how are you planning to train for this, or how are you gonna try to detect those?
So we thought it would be really easy, but we had some meetings with more experts in training models, like Winston Tim, for example, and they explained to us that it is a lot more difficult than we thought it would be, because of a lot of different reasons that are beyond my expertise to explain to you all, but we're working with collaborators basically, and so we have to have the right samples to be able to train the models to be able to learn to detect hydroxy, and getting the right samples is really hard for hydroxymethylation. So you can get nucleotides that are hydroxymethylated, but to incorporate them in a way that you expect is not as easy as we would like. So it's more of a chemistry problem than anything, a biochemistry problem. But yeah, so we've got to get the right samples. So in answer to your question, the samples is what we're looking for. So we think we can do it for specific, like very short, like motifs, for example, but for full whole genome hydroxymethylation calling, it may be a while before we're able to do that.
Yeah, I think that answers the question. Thank you very much.
Does that answer your question? Okay.
Yes, it did. You got the thumbs up. Cool. Thank you very much for getting up early, talking with us. That was very informative.
Problem.
All right, thank you very much for the opportunity to present what I've been working on. I'm Wouter, and I'm a postdoc bioinformatician and working with long-read sequencing data. I'm also the developer of Nanoplot and other tools for QC of nanopore datasets. But today I'm happy to present some methods I've been working on to visualize and analyze modified nucleotides from nanopore sequencing. And it's a valuable feature of nanopore sequencing that if you don't use any amplification in your library preparation, you also get theoretically all nucleotide modifications. There's a nice recent review on this subject, so if you want to get up to date on the methods for identifying modifications, then that would be a good start. The first tool developed for this and the most often used is Nanopolish, which is convenient, works well, but I'm also very excited about the methylation-aware base calling, which would directly identify modified nucleotides by explicitly taking them into account while base calling. Something that we badly need is a file format for modifications to store the nucleotide modifications and scale well to hundreds of modifications in an easy computer readable format. The things I'm presenting today are available on GitHub, and our preprint is on bioRxiv since last week. The main purpose of my tool is a genome browser in a dynamic HTML format, allowing you to zoom and select the most interesting bits. I'll get to some examples after the slides. You can show both the raw per position probability of having a nucleotide modification per read, or you can get the frequency of having modification at that position and annotate these with features from a GTF or BED file. The tool should be easily installable from Bioconda or PyPy and builds on excellent modules from the Python data science ecosystem, PyRanges, PySam to work with bioinformatics files, and Plotly for visualization. For further convenience, I've also included a SnakeMake workflow, which can run Nanopolish for multiple samples and multiple regions of interest. This is the first example, so it's the actin gene in this case, and it will show you in the top part the frequency of having a methylated CpG for all reads. The central part shows you per read the likelihood, the probability of that this certain CpG is methylated or not methylated, with in blue the unmethylated, and within orange like the more methylated nucleotides. And the bottom part shows you the annotation. Here is the gene annotation, and the gray bars in this case are DNA's hypersensitivity regions. Another example is a highly expressed gene in these cell lines, which shows you that, as you would expect, the regulatory regions in front of the gene are unmethylated, while the rest is to some extent methylated, with the exception of here, again, another unmethylated position, which is again a regulatory feature. You can visualize multiple samples, either like this, overlaid traces, or like this, with all traces separately, which at the same time shows you that calling methylation is quite reproducible across samples, provided of course that you're working on the same tissue, as you would biologically expect. I've included some basic quality control plots to give you a bird's eye overview of the data you're working on, and allowing you to spot larger, presumably technical differences between your samples of interest. So you will, for example, get the number of positions for which modifications were called. And as you can see here, the S10 is a Promethean run which did not perform so well. It was on dying lymphocytes, if I recall correctly, while the S12 is a combination of 5 Promethean runs. And the impact of this difference in coverage will also be visible in the rest of the plots. Here you can see the distribution of the frequency of methylation across the entire set that you're plotting in this case. And as you can see, all those from S10, the orange yellowish bars, show a less smooth pattern. It shows you far more discrete positions because the coverage is lower. Also, the estimation of the methylated frequency is less accurate, and the other higher coverage samples give you a more smoother distribution. This plot shows you roughly the same information as the previous plot, but represented differently, which will tell you that most of the genome is methylated around 75–70%, with a range for standard deviation between 50 and slightly above 80. And here you could also already spot some differences, that the S7 is on average methylated less. And you could wonder if that's a biological or a technical difference. And I don't have an answer to that right now. What is also produced are pairwise per position correlation plots, like this one. And here each sample is plotted against each other sample, and samples which are more similar will show a better correlation. And as such, you can also identify outliers like the S4 here, which is a different cell line, well, a different cell type. So naturally, it does not correlate as well with the other samples, but it's at the same time a nice sanity check that we can spot this on this level already. You could do the same plot for larger data sets. Here is an outlier, and even larger data sets with another outlier here. And you can see that here that other samples correlate very nicely, while this one is all over the place. I've included some basic statistical tests in my package. They work, but some refinements are necessary, and it's still a work in progress. After phasing reads with WhatsApp, you can quantify the allele-specific methylation or modification, which can then be used to find parental imprinting and also X chromosome silencing. It uses a simple Fisher exact test, a method which can also be expanded to differential modification calling across samples, but this could use a better statistical approach to proper take within group heterogeneity into account. And I'll show you some examples of these. So this is a region where one haplotype, I'm not sure if it's the maternal or the paternal, but it's just phased. One is mom, one is dad, shows a strikingly different methylation pattern. So you can see here there's this gene, it's the GNAS antisense gene, and that the promoter region is differently methylated. So in one phase it's highly methylated, and the other one suddenly is not methylated anymore. If I would zoom in on this one, it would look like this. So you have one phase with the almost unmethylated region, and the other one which is partially methylated, partially unmethylated. Zooming further in will give you an even finer understanding of this. It shows you here the individual CpG positions and their likelihood of being methylated. So again, this phase, these chromosomes are in blue, meaning that they're not methylated, while these are silenced. This is another example of a known imprinted gene in which you see a large difference between one chromosome and the other. So here there's strong methylation in phase 1 and less methylation in phase 2, with especially a large difference here in the center, which is on a single exon non-coding RNA. So that looks like some interesting biology. These regions are known to be imprinted, so this is at the same time a validation of what we're finding, but we could also use this to find novel regions. And we can also use this to look at X chromosome silencing, because the sample we're working on is female. And then it looks like this. So here you have a promoter, a bidirectional promoter, which in one phase is completely silent, and in the other one is completely open to transcribe those genes. So that's essentially what I wanted to tell you today. You can find my tool on GitHub, our preprints on bioRxiv, and me on Twitter, and I'm happy to take your questions. Thank you for your attention and have a nice day.
Thank you. Questions? I think it's really nice, and I would have liked to have this like 2 years ago when my student did the methylation. I think she would have gotten a better grade with all these nice pictures. So can you also use this not only for nano polish, but also other modification callers like Tombow, or I think there was 1 or 2 others recently?
Yeah, there are about 10 tools at the moment. It's currently specifically for nanopolish, but I'm totally willing to make it possible to add other input modes. But right now, I can't predict which of these tools is going to survive, which is going to be the dominant, and which file format is going to be convenient. It seems that Tombow stores everything in the FAST5 at the moment. Also, the methylation-aware base calling puts its methylation probabilities in the FAST5 format, and I'm not really looking forward to parse these for Promethean runs. So that's why I'm first waiting for the community to develop an easier format.
Cool. Any other questions, comments? No, I think we're all pretty fried. I think it was really nice, and also thanks for writing nanoplot. I think it's very useful, and I think we will go through this tomorrow.
All right, great.
Cool. Thank you.